So yeah, today I'm going to share about the role of synthetic biology in cancer treatment. So the recent completion of human genome sequencing is an exciting news, but actually since long ago, scientists can already manipulate different living cells for our own use. And this field of science is called synthetic biology, where living cells can be programmed to obtain new abilities by the insertion of artificial gene circuits. And in the context of cancer treatment, synthetic biology is usually used to produce cancer vaccines. Unlike the vaccines for other infectious diseases, cancer vaccines aim to train our immune system to recognize the tumor cells so they can fight against the malignant cells specifically. So there are actually two main directions of synthetic biology that can be applied in cancer treatment. So first of all, the scientists can design a system to deliver toxins specifically to cancer cells, or they can boost the adaptive immunity against the cancer cells so that the vaccine will not only be able to treat the existing cancers, but also can hopefully prevent the malignancies if sufficient memory immunity can be elicited. So today I'm going to share two examples of how living cells can be manipulated to kill cancer cells with high specificity. So the first one is about bacteria. So bacteria is actually a common agent modified by scientists, as it is very convenient for us to introduce plasmids and instruct them to carry different functions. And bacteria actually tends to stay within the solid cancers, as the tumor microenvironment create a very good condition for these pathogens to evade immunity. Meanwhile, the bacteria can communicate with each other using small molecules. And this process is called quorum sensing. So this quorum sensing is actually, this feature was actually recently utilized by a team from the University of California to create a synchronized lysis circuit in Salmonella. So this one is the first example, the synchronized lysis circuit. So the bacteria was modified to produce anti-cancer drugs. And the Salmonella bacteria was also designed to burst when they reach a certain population density. And in the end of each cycle, a small percentage of bacteria will remain alive, and they will continue to grow to replenish the colony. And then the kill switch will be triggered again when the bacteria reaches a certain density. So the drugs can be released periodically. So this lysis circuit consists of several plasmids that are controlled by the same promoter, LAX-L. So first of all, the bacteria can produce an auto-inducer protein, AHL, under the control of the LAX-L promoter. So the AHL protein can actually diffuse out of the bacteria to communicate with the nearby bacteria. And the AHL concentration continues to grow, continues to increase as the population grows. And the AHL protein can bind to the LAX receptor and trigger the expression of more AHL protein and also the therapeutic drugs. And when the AHL reaches a certain threshold, the bacterial lysis protein E will be promoted for expression, and then the bacteria will burst to release the drugs. So let's see if it works. It does, okay. So this one is a video showing how the system works. So you can see the bacteria growing, and then when they reach a critical threshold, it's gonna burst and release the contents inside as shown in the blue color, right? Okay. So the team has verified the engineered bacteria can lyse periodically. So then the team engineered the bacteria to express three proteins. The first one is the hemolysin E protein. It is a pore-forming anti-tumor toxin. And the second one is CCD, which can trigger tumor cell apoptosis. And the third one is CCL21, which can be used to activate the host immune response. And then the team tried to treat the subcutaneous tumors with the bacteria, and it was shown that the intratumoral delivery of the synchronized lysis anti-cancer drug-producing bacteria can actually significantly reduce the tumor growth, as shown here. So the team then tested the engineered bacteria in an experimental model of colorectal metastasis within the liver. So the bacteria was shown to be able to efficiently colonize within the tumor and occupy the avascular tumor compartments after the oral administration. And these engineered bacteria can hence carry drugs to the necrotic core of the tumor, which would actually usually be quite hard to be targeted by standard chemotherapy, which is more efficient against vascularized regions. So while combined with the chemotherapy, the oral delivery of the bacterial therapy actually led to a marked decrease in tumor activity over the period of 18 days. So from this example, we can see how bacteria can be manipulated to carry drugs and released periodically within the tumor to kill the cancer cells. So the second example that I'm going to share is a cancer vaccine that is based on filamentous phages. So phage is a type of virus that only affects bacteria. And due to their antibacterial properties, bacteriophages has actually been approved to be administered in meat and poultry products as food additives by the FDA since 2006. And in 1985, Josh Smith has actually described a phage display technology where target peptides can be expressed on the surface of the filamentous phage by just inserting DNA sequences into the phage genome. And it was later found that the phage can display different peptides on their surface proteins like P3, P7, and P8. And this technology enables easy editing of the phage capsid protein, after which engineered phages can be manufactured in a large scale with a relatively low cost compared to nucleic acid and protein drugs. So as a foreign antigen, the phage particles can be recognized by our immune system when they're administered intravenously. And the phage can also trigger both B and T cell actions, and also they can be rapidly removed by the reticuloendothelial macrophages due to the non-specific adsorption of the circulating fibrinogen, immunoglobulins, and complement factors. So in 2005, a team engineered the phage to display an antigenic peptide taken from the melanoma antigen gene protein, which is called MAGE. And then this sequence is displayed on a P8 protein on the phage. So this peptide is a cancer-specific protein that is not present in a normal tissue. So when displayed on a phage, the cancer cells can be targeted specifically. So interestingly, both the wild type and the engineered phage can increase the NK cell activities. And in both preventive and treatment experiments, the hybrid phage can induce strong production, reduce tumor growth, and also improve the survival rate. Meanwhile, the splenocytes isolated from the mouse immunized by the fusion phage can also lyse the B16 melanoma cells, demonstrating that the engineered phage can serve as a potential vaccine for immunotherapy. Well, on the other hand, as a very common target investigated by cancer therapy, HER2 antigens have also been displayed on phage surface for evaluation as a possible cancer vaccine. So a group has actually isolated immunogenic HER2-derived peptide, which is called E75, and displayed them on lambda phages, which is a type of phage. And although the tumor grows in both the control and treatment group, you can see there's a significant increase in the level of IL-4, interferon gamma, and also CD8 positive cells in the immunized group, while leading to a reduced tumor growth. However, unlike the previous case, the phage injection did not provide protection to the mice against the cancer cells. And it was also noticed that the regulatory T helper cells were induced by the phage vaccine, which may hamper the T cell activities against the tumor. Nevertheless, as shown in these two examples, mice receiving phage as a cancer treatment or preventive vaccine is still a possible treatment for cancer, actually. But then it's still subjected to further research on how to improve the efficiency of phage-based cancer vaccine, while it is also worthwhile looking into the possibility of modifying phages to a preventive vaccine for cancer. Well, so the current cancer treatments are mainly cytotoxic drugs or antibodies, and it is very difficult for us to target the cancer cells with high specificity. Nevertheless, with synthetic biology, it is still hopeful for us to improve the specificity and also the efficiency of cancer treatments by modifying different living organisms to target the cancer cells. Right, so this is my presentation, and if you're interested, you can take a look at these papers. Right, yeah, okay, that's it from me.